Hello everyone, my name is Simin Tabasi, and during my presentation I will discuss about most recurring deficiencies we encounter during oral solid drug product quality assessment. Due to time limit, in order to better focus on the objectives of this presentation, I will assume majority of attendees are familiar with the basics of ANDA submission. The next few slides are messages from OPQ. Since previous speakers have presented them, I will only click through them. This presentation is intended to provide you with some of those most common deficiencies that impacts review cycles and drug approval. Due to time limit, I will cover some of deficiencies related to setting acceptance criteria for drug product description, drug product assay, and organic impurities, only those covered by ICH Q3A, Q3B, and drug product specific USP monograph. Second, I will also discuss some common deficiencies in drug product stability. For each of above section, I will provide points to consider when responding to deficiency letters. And finally, I will provide a case study with 2 challenge questions. The acceptance criteria for appearance of dosage form is product specific. It is a qualitative description of dosage form based on final appearance, such as shape, size, dimension, color, engraving, scoring, and overall integrity. The most common failure is excluding acceptance criteria for product integrity or defect in release and regulatory specification. In such case, below OPQ general deficiency language is cited. Some applicants may respond that tablet or capsule defects are being tested during manufacturing process, so it is not necessary to include it in product specification. Applicants should consider that in-process evaluations of defect in tablet or capsule do not address the changes that may happen due to warehousing, packaging, as well as instability during shelf life. In this section, acceptance criteria for assay of all drug product is commonly 90 to 110% of label claim. However, in case of narrow therapeutic drug product, the assay limit is 95 to 105% of label claim. Based on ICHQ-6A, the following deficiency may be cited. Applicants should consider that based on ICHQ-6A, as a control, the agency may recommend a tighter limit for release specification to ensure quality during drug product shelf life. In this section, the product specification may include the following list of impurities: specified identified, unspecified unidentified degradation products, any unspecified degradation products, and also total degradation products. Degradation products which should be controlled in drug product specification are: first, metabolites, which evaluation of pharm tox, clinical, etc. is required. Second, impurities from degradation of drug substance, as well as those impurities formed during manufacturing and storage of drug product. Additionally, impurities formed due to interaction between drug substance and other components of drug products, such as excipients, container closure, etc., must be controlled. The basis for setting limit is: first, drug product USB monograph recommendation, if exists, which is only applicable to specified identified degradation products. Second, ICH Q3B recommendations, in which a specified identified degradation product should not exceed qualification threshold. The specified unidentified degradation product should not exceed identification threshold. Any unspecified degradation products, regardless of being identified, or unidentified should not exceed identification threshold. One of the most common deficiencies is including those process impurities of drug substance which are non-degradation product in drug product specification at ICH-Q3B qualification threshold. Controlling these impurities of drug substance in drug product specification is not necessary, since these impurities will not increase during shelf life of drug product. If any applicant wishes to include them in drug product specification, it should remain within ICH-Q3A limit. Another common deficiency is that the limit for any unspecified degradation product is above ICH-Q3B identification level. Next is failure to identify and control all known degradation products which may potentially form in the drug product. Below is an example of deficiency language which is cited when such information is missing. Applicants are recommended to access publicly available information on approved drug products, scientific reports, literature, etc., to ensure that relevant degradation products are controlled and methods are adequately validated. Specified degradation products with a limit above qualification threshold is considered acceptable if supported by the following justifications. First, USB monograph recommends a limit above qualification threshold, or supporting data from non-clinical studies, publicly available information, or comparison to the RLD using the same method of analysis. Common deficiencies are, first, failure is justified proposed limit of higher than qualification threshold for degradation product based on the above criteria. I would like to mention that QSAR is a useful tool for prediction of toxicity of an individual degradation product, but generally is not conclusive for qualification purposes. Next is failure to justify the degradation product is a human metabolite. This is a case when firm sets the limit above qualification threshold and claims that the impurity is metabolite without justifying by non-clinical studies, publicly available information, or toxicology studies. Failure to adequately compare the impurity in endo product to that of RLD is another one which is very common. As a prerequisite, applicants are expected first to demonstrate that the degradation product of interest in endo and RLD are in fact the same, using specific methods to identify and characterize the degradation product. Simply comparing retention times of impurities in endo and RLD products instead of identification and characterization is not acceptable. Unless described in product-specific USB monograph, the total degradation products is the sum of acceptance criteria for individual specified, identified or unidentified degradation products. Applicants should consider that significant human metabolites should be excluded from the sum of total degradation products. The limit for the sum of all impurities, including metabolites, should not compromise the assay potency of drug product through shelf life. One of the most common deficiencies is a failure to justify proposed limit for total degradation products. Below is OPQ general deficiency language that perhaps many of you have received. Based on ICH-Q1A, a minimum of 3 submission batches should be placed under stability conditions and provide the stability results of samples under the following conditions in the applications. 12 months under control room temperature, 6 months under accelerated condition, and in case of significant changes in samples under accelerated condition, 12 months stability under intermediate conditions should be provided. Common deficiencies. First, insufficient stability duration to support 24 months expiry. For example, the proposed expiry is based on 6 months accelerated and 6 months long-term stability, which is not acceptable. Next is the proposed expiry is based on a 6 months accelerated and 12 months long-term stability, but significant changes in product quality during 6 months accelerated condition occurred. Firm did not provide intermediate results. Another one is significant changes happened in product quality during first 3 months of accelerated condition. In such case, the expiry will be based on duration of long-term stability. In all above cases, we refer all applicants to ICHQ-1E for calculation of expiry. Another very common deficiency is lack of mass balance in stability data for assay versus total impurities. For example, noticeable decreasing trend in assay, but total impurities remained unchanged. Please keep in mind that although during method validation and force degradation studies, mass balance between assay and impurity methods may have been established, but that cannot be used to undermine lack of mass balance in stability results of assay and total impurities. Next is lack of justification and investigation for out-of-trend and out-of-specification results. One example is firm reports out-of-specification level of unidentified degradation product without being investigated. The justification, such as it was a one-time occurrence or the degradation product could not be isolated and investigated, adds more to assessment cycle. Lack of stability results for reconstitution of drug product per labeling is another one. For example, the labeling recommends that the product be dispersed in a juice or water and administered to children or infants. In such case, based on a labeling, orally equivalency studies such as crushing, suspending, viscosity, dose recovery by a spoon and dropper will be requested. Additionally, depending on recommended storage time, whether it's a 1 week, 1 month, for example, a full stability results per specification at varying time points will be requested. And finally, lack of data to support the stability of solid state at release and during shelf life is another one. The stability of solid state is requested when the rock substance is low soluble, BCS class 1 and or 4. The solid state of drug substance thermodynamically is not the most stable. Manufacturing process may result in a change in a polymer such as wet granulation, dissolving API in solvent. And finally, firm also should consider that conversion in crystal shape, for example, to needle shape when applicable, may impact the processability of drug product. In conclusion, I would like to emphasize that in addition to numerous ICH guidance, the US FDA has issued many guideline documents and literature papers. It is responsibility of applicants to learn, understand, adopt, and apply the regulatory expectations before launching drug product development, manufacturing, preparing, and submitting ANDA applications. Although filing an ANDA is a significant challenge, applicants should first focus on the end goal, developing and manufacturing of safe, potent, and effective drug product, followed by receiving FDA approval. I would like to express my gratitude to Dr. Rosenkrantz, Dr. Saeed, and Dr. Gill for their support and encouragement. Before I close my talks, I would like to present you 2 questions. This is a case of narrow therapeutic drug product. MDD is 2 mg and the assay is 95 to 105%. The acceptance criteria for impurities are in line with ICHQ-3B recommendations. Impurity Y and impurity Z are both specified identified degradation product and the limit for each is no more than 1%. Impurity at RRTX is a specified unidentified degradation product with a limit of no more than 0.5%. Any unspecified degradation product is no more than 0.5%. Based on this presentation, is the overall acceptance criteria acceptable? I give you a few seconds for response. Yes, the overall acceptance criteria for organic impurities are acceptable. However, please keep in mind that the overall impurity profile, I mean sum of all impurities, for this product is about 3% and the assay limit is 95 to 105%. If impurity levels in a releasing batch is at or close to the specification level, there is a high potential for assay failure during shelf life. For the same product, firm proposes a limit of no more than 3.5% for total degradation products. Based on this presentation, the limit for total degradation products should be no more than 3.5%, 3%, 2.5%, or 2%. I'll give you a few seconds to respond. The proposed limit for total degradation products of no more than 3.5% is not acceptable. It should be no more than 2.5%. Total degradation products, as described in MAP 50172, is sum of individual specified, identified, and unidentified degradation products. With that, I would like to thank you all for attending this presentation.
And thank you, Simin. Oh, we're getting our audio reconnected here. Thank you, Simin. This is Jeff. We're just going to go ahead and switch the layouts here a little bit to get ready for our next presenter. And the AE, if you're all set, I'm going to go ahead and unmute you. You should be able to hit that. Right there. Go ahead and accept. And you should be able to take control now, AE, and I will turn it over to you.
Hi everyone. The title of my presentation today is Common CMC Issues for Manufacturing Process and Facility Assessment. In this presentation, I'm going to discuss the integrated process and facility assessment in our office, explain the review expectations, and also examine common process-related.
Hey, I apologize. I apologize for interrupting. This is Jeff. We're having just a little bit of trouble hearing you. So what we want to do is check real quick the microphone icon up at the top of your screen. You see the green mic icon. If you're using are you using a headset or speaking directly to the laptop? Oh, and now, you're muted. Okay, let me unmute you.
Yes. So can you hear me now?
Not very well. Are you using a headset or are you speaking directly to the laptop? And
I'm speaking directly through my laptop. Is this okay now?
Actually, what I'll ask you to do is a dial-in number.
Okay, so I'm going to use the dialing number.
And thank you for your patience, everybody. We were having a little bit of issue. Phone. We are going to get her dialed into the.
Hello? Can you hear me now?
Yes, we can. You should be able to listen and speak on your phone alone. So will you go ahead?
I tried to mute my microphone, but I don't know.
Are you dialed in on your cell phone, Pei?
Yes, I'm dialing with my cell phone.
Okay.
Okay,
Could you
how
please
is it now?
turn down the speakers on your computer?
Hello?
Hello, we can hear you great. Could you please turn down the speakers on your computer?
Oh, yes. Okay, I just did.
Okay. And now if you'll just speak a little bit louder into the cell phone, we should be all set to go.
Okay. So in this presentation, discuss the integrated process and facility assessment in our office, explain the review expectations, and also examine common process-related deficiencies. The Office of Pharmaceutical Manufacturing Assessment, or OPMA, is a sub-office in OPQ. We perform evaluations of pharmaceutical manufacturing by integrating the assessment of manufacturing process, facilities, and pre-approval inspection. An OPMA reviewer provides a holistic manufacturing assessment of the process, facility, and microbiology information for solid oral dosage forms. In this way, a more informed and risk-based decision can be made. This slide illustrates the holistic manufacturing assessment process. During the assessment review, the assessor will look at the adequacy of process understanding and also to determine if critical controls are in place and well documented in the application. And if it's determined that pre-approval inspection is needed, then the assessor will participate the pre-approval inspection with an ORA officer to determine that whether the facility is capable of implementing the listed manufacturing operations in conformance to CGMP, and also to see if there's any data integrity concern for this application. The purpose of manufacturing assessment to evaluate the adequacy of final commercial scale manufacturing and process. We're using a risk-based concept to provide the assessment. A robust manufacturing process with a well-defined control strategy should be developed so that the drug product produced will have the consistent quality.
So assessor would look at the manufacturing process development report in module 3.2.22, and adequate process and product understanding should be demonstrated. In addition, a risk-based approach for the manufacturing process should be implemented. The assessor also look at the executed batch record, and this is required by 21 CFR Part 314, and the batch record, executed batch record should be included in 3.2.R. In addition, the executed batch records establish the baseline for the manufacturing process and control for the commercial batches. The assessor also examines the commercial batch record or the master batch record. In the master batch record, a batch formula, the manufacturing process should be clearly defined, and also equipment, process parameter ranges, and also in-process control should be documented in the master batch record. In the next couple of slides, I'm going to discuss 10 common process-related deficiencies. The first one is related to the API polymorph. It is documented that the manufacturing process could be harsh and create additional stress to the API, and this will in turn promote the polymorph conversion. So if the risk for potential polymorph conversion is identified, additional data such as XRD or the IR data should be provided to demonstrate that there is no change in the API morphic form during processing, and also account storage. In addition, the suitability of the analytical method should also be demonstrated. If there's any polymorph change identified, the effect of these potential changes on the in vivo and in vitro performance of the drug product should be discussed. Blend uniformity and stratified content uniformities are very important studies to demonstrate that the produced drug product will be of consistent quality. For your BU study, process development data should be provided to justify the BU sampling plan and the acceptance criteria. In addition, if you are going to propose a larger BU sampling size to meet the BU acceptance criteria, additional justification should be made. And also, for a high-risk product, stratified BU sampling plan and acceptance criteria may also be needed for the commercial batch manufacturing. The third example is related to lack of supporting process development data to justify the critical process parameters and in-process control. If you are proposing a wide range of the weighting control for a functional coating process, it may not be acceptable. Additional data should be provided to justify the weight range control for this process. The same thing applies to IPC for the granule size and the moisture content. Process development data should be provided to justify the proposed acceptance criteria. Many times the proposed commercial batch size is different from the executed batch size. In this case, the differences in the equipment percentage utilization should be discussed. And also, if there's any difference in the process as it relates to the proposed process parameters, such as the blending time versus the blending rotation speed, should also be justified. And also, for the master batch record, clear instructions to the operator should be provided in the master batch record. Oftentimes, we see a subjective manufacturing process description. For example, for high shear granulation process, the statement would say, if necessary, add additional amount of granulation fluid. This does not provide a clear instruction to the operator. So, in consideration for the IR response, you should provide process development data to justify the amount of granulation fluid, rate of binder addition, and wet masking time for this critical high shear granulation process. We also recommend you to establish acceptable granulation endpoints such as the impeller torque and the power consumption, so that the granulation endpoint can be clearly defined. Production yield is also an indication of the robustness of your manufacturing process. If no low yield is noted, the root cause of the low yield should be properly investigated or justified. For example, is it caused by additional sampling, or is it because the batch size is small, which caused the low yield? If process issues are identified, then the mitigation strategies to ensure a robust manufacturing process should be provided in the application. The drug product intermediate hold times and conditions should be identified and justified with additional adequate data. The test that's used to conduct to justify the hold times and conditions should address the quality issues. For example, if you have a low-dose drug, then just providing the assay results for the final blend to justify your hold time may not be sufficient. We would like to see the BU data as well. And in addition, if you're proposing a hold time for the granulation fluid, then microbial limits may need to be provided. So considerations for the IR responses to the hold time deficiency, identify the high-risk intermediates and also discuss CQAs that may be affected by holding the intermediate for extended period of time. And also CQA data should be provided to support the proposed hold time. Regarding the process parameters for the commercial batches, we would like to see ranges, process parameter ranges specified and also justified with either established scale-up principles or based on your development experience. As I said earlier, we're looking for commercial readiness. So providing those data is a way to demonstrate that you are understanding, the critical process parameters and what's required to manufacture your product. And sometimes the applicant would propose to use sublots for the commercial batches. And one of the common deficiency is inadequate IPC control for each sublot. In the IR response, please provide sufficient material characterization and process controls for each sublot before combining to ensure that variation in sublots is not masked by subsequent unit operations. And the last example is about equipment compatibility. If you have a liquid or semi-solid formulation and the product contains co-solvents or surfactants, then leachable or extractable study for the polymeric manufacturing components such as tubings or filters should be considered and provided. You can consider to use USP 1663 and also USP 1664 principles and general approaches to demonstrate that all formulation contacting polymeric components are not reactive, additive, or absorptive to alter the safety and quality of the drug product. I want to say that over the last few years, the quality of submissions we received has greatly improved. And avoiding the 10 most common deficiencies that I just discussed will further improve the quality of your application. The agency used a risk analysis to make process and control decisions. We recommend continued use of QBD principles in establishing material controls, CPP, IPCs, and finished product acceptable criteria. The applicant should prepare their application with adequate data and discussion to demonstrate that there is a good process and product understanding. The applicant should verify that the commercial batch records include clear instructions for the operator, and also the process parameter ranges and also in-process controls are well justified. If the proposed commercial process deviates from the development or the executed batches, an explanation should be provided. And also, if the proposed commercial process parameters and controls deviate from the development or the exited batches, adequate justification should be provided. I would like to thank my OPMA colleagues, Mahesh, Rocky, and Rapti, for their help in preparing this presentation. Now let's go to a challenging question. The first one is, which office in Cedar performs evaluations of pharmaceutical manufacturing by integrating the assessment of the manufacturing process, facilities, and pre-approval inspection? The correct answer is OPMA. Question number 2. Select the true statement. A, the purpose of manufacturing review is to evaluate the adequacy of final commercial scale manufacturing process and facilities. B, FDA conducts risk-based evaluation for incoming applications. C, a robust commercial manufacturing process with a well-defined control strategy that produce drug product of consistent quality is not required for drug application. D, all of the above. So the correct answer is A and B. So this concludes my presentation. Thank you.
Thank you, Pei. That was very well done, and thank you so much for rolling so effectively there with the little snafu we had with getting your audio connected. You can just stay on your cell phone. You can go ahead and mute that phone because that's how you'll communicate with us during Q&A. Speaking of Q&A, our first speaker, Simin, should be joining on the phone bridge as well.
Simin, are you on the line? Just want to make sure.
A few questions come in, so even though we're running a little bit behind schedule, we're going to go ahead and
Our first question for you is that if the pharmacopoeia has a total limit of NMT 2.0% and the MAP's recommendation limit should be NMT 1, what do you recommend?
Yes. Yes, I am.
Okay, thank you. Our next question is about if a reference listed drug is showing an increase in impurity, can that be used or taken as a reference to justify an ANDA application?
USP limit. If it's higher than ICH Q3B limit, then we go first with the USP limit.
Okay, great. Again, another question for you. Do companies need to complete accelerated stability studies if the product fails specification before the last scheduled time point?
And provide that information to the agency that this impurity is the same as the one in the reference product?
Sure.
We will take a
Do
look
companies
at the
need to complete an accelerated stability study or studies if the product fails specification before the last scheduled time point.
I'm sorry, repeat it again. Okay, if company fails the accelerated study within any month during 6 months accelerated, they should complete the accelerated by
Great.
6 months.
One more question for Simon, and we'll go to
to understand better about
so the question is, for drug product total impurities, is this based only on specified impurities, or should they be including any unspecified, unidentified impurities in the total?
Study per protocol, regardless they fail or pass. It is only based on specified impurities, whether it's identified or unidentified. If firms are observing some particular unidentified, unspecified impurities or increasing, then I would suggest them, if they are getting close to identification threshold, I would suggest applicants to identify or list them as an
Okay, great. Thank you. Our next question is for Pei. So are there any guidances describing the maximum processing time for the manufacturer of submission sets and of batches?
And this way they can control these particular impurities during the stability of the product too. And that can be added to the total impurities. Well, currently there is no maximum processing time for manufacturing of batches, but you have to keep in mind that the product shelf life is determined based on the first API is introduced to the formulation. So if you have a very long manufacturing time, then you're losing your shelf life claim. Or you have to provide extra long shelf life in order to fit into the shelf life criteria.
Okay, great. Another question for you.
Sufficient data should be
what
provided
principle
to
does FDA usually decide to evaluate
data
CPP?
to support the whole time, and also keep in mind that your shelf life of the product starts when the API is introduced to your excipient. Well, I think we use a risk-based approach and determine that whether variation in those process parameters would have significant impact on the product quality. So, for example, if you have a high water-soluble drug substance and you're trying to dissolve it in water, and maybe the blending time may not be that critical. However, if you have a low solubility compound,
and we have another question about risk for you.
Or make
Can you please describe how the agency would consider high-risk intermediates? For example, you listed them on slide 15 and 16.
That the product quality can be met. So it really depends on risk. Well, I think for that particular slide, I was referring to the final blend, because many times we see that the manufacturer would, make the blend, and then the next, whether it's the encapsulation step or the tableting step, it may be either performed, later on or at a different facility. So in that case, it will be considered high-risk intermediate. And also, it depends on the drug loading. So typically, we are concerned with the stability of your intermediate. However, if you have a low dose
Wonderful. Thank
product, and then the
another
drug
question
loading
for
will be
you.
very low. So
You mentioned that
case,
microbial limits should be provided for binder
data,
solutions in some cases. If the dosage form is solid oral dosage form, is the solid oral dosage form where the microbial risk is low and release testing had adequate microbial specification, is it still necessary to control for a microbial limit for a binder solution?
Well, if you're proposing extended hold time for the binder solution, and many times the solution may contain sugars or some carbohydrates, which is
Great. Thank you so much. Right, we're going to go back over to Simon for a question.
So in that case,
please explain your case study on slide 14. If the applicant has 12 months of long-term stability without significant change on accelerated, are they able to request 24 months shelf life? Okay, great. Thank you. Another question for you. Is the agency expecting that a reconstituted product, should that be tested in all conditions as mentioned in the label, or can they just generate data for the worst case condition within the labeling recommendations?
Yes, they can request 24 months shelf life. I would, depends on the labeling. If labeling says that the product is stable in refrigerator.
All right, great,
and
thank you.
For
we
example,
have another question, and hopefully I'll read it correctly.
I would recommend
is it mandatory for, it's a little confusing for me, but is it mandatory for ANDA batches packaged for bulk product into the final marketing container that can it be performed within 30 days?
Then perform at varying time points.
Oh, yeah, yeah. Correct, thank you. Is it mandatory for ANDA batches packaged in bulk product into the final marketing container? Can it be performed within 30 days?
I believe this question is for pay, and this is about bulk packaging, if I don't mistake. So can you repeat the question again? Well, I guess each company has their own SOP, and as I mentioned earlier, if there is a risk, if you don't package that immediately, and if it's going to have potential risk for the product,
and we have one last question for
justification and specify
the
limit.
BU sampling plan as per ASRM, is that acceptable?
No such risk is identified, and then you can just incorporate into your manufacturing process.
Is the BU sampling plan
really,
per the ASRM, is that an acceptable one?
Can you repeat the question again? The BU sampling? Usually for BU sampling plan, we like to see that the sample size and also the locations are justified, and usually the sample size should not be greater than 3 times the unit dose of your finished product. However, we do recognize that sometimes for
Lisa,
a very low dose, and you may need to go above the 3x, and in that case, additional justification needs to be provided to demonstrate that your
we are now
blend
going
is
to go
homogeneous.
ahead and move on to the next session, which is going to be presented.